Hey, do you ever wonder mid-rep how exactly all this works? Like if your whole body, even your muscles, are made up of just trillions of tiny cells, how do they all work together to lift all this weight? Your strength actually comes down to an incredible kind of modular precision. All of your muscles across your whole body are, in a way, millions and millions of repeats of the same handful of structures. By going down to the molecular level and building up the mechanism of your muscles, protein by protein, you can get an incredible view of the sheer coordination it takes just to keep you up and moving. The gears and levers that make up your muscle fibers trace their history back billions of years, before life was even multicellular. By remixing a few structural proteins and assembling them in just the right way, our cells can accomplish incredible amounts of work with very little input. But let's just get right into it. Crack some creatine and settle in for this one, folks. Let's start exploring the sarcomere. And hey, welcome back to Clockwork, a show about the tiny, beautiful molecules at the foundation of everything you do. Make sure you subscribe if you want a better understanding of how your chemistry roots you to the world. And check out our community on Patreon to see our latest sticker packs, temporary tattoos, and papercraft molecules that celebrate your biochemistry. But anyway, let's get right into how your muscles work. And I'm gonna do the best I can to speedrun through the anatomy to get to the cool stuff at the molecular level. So yeah, the basics. All of our motion is powered by our muscles, whose main job is to contract and get shorter. Every movement comes from muscle tissue pulling on some part of your skeleton, with most muscles coming in pairs so one partner can pull the other one back after a contraction. Moving further down, that contraction requires an incredible amount of coordination to make sure opposing forces don't accidentally rip your cells apart or anything. So muscles are made of very unique cells called myocytes. You normally imagine cells that look like this, right? They're very square or very round, but always like roughly the same length and height. Muscle cells are some of the most specialized tissue known to nature, and they only care about getting shorter and longer. Therefore, these cells are long, thin fibers that are the result of lots of progenitor cells fusing together, which means that one muscle cell actually has several nuclei running the show here in tandem. Underneath the surface, these cells are just bundles of protein fibers called myofibrils, which are surrounded by support structures like transverse tubules and the sarcoplasmic reticulum, with most of the rest of the space in here getting absolutely stuffed with as many mitochondria as possible. You know, to power all the movement that happens in here. But now we're getting to the good stuff. So if we briefly switch to an electron microscope view of these myofibrils, you're going to start understanding what makes these cells so powerful. Each myofibril is incredibly well organized. I mean, look at these bands of protein. They line up almost perfectly across several different myofibrils. That's because each fiber inside every muscle cell is just the same fundamental unit repeated over and over. This region, dark band to light band to dark again, is a single structure called a sarcomere. Across a single muscle, every sarcomere is pretty much exactly the same. But different muscles and muscle types might have slightly different sarcomeres with slightly different sizes. Either way, as your muscles grow, they're basically just stacking up new sarcomeres. So if you learn how one single sarcomere operates, you basically know how the entire muscle works. So if we zoom all the way in, we can finally start exploring the proteins that power all this movement. Behold, the sarcomere. Down here at the molecular level, it honestly feels like we're in a dense forest more than anything, with long, thin filaments and elastic connectors towering over us. A sarcomere is three main components repeated over and over again, basically. We've got the thick filaments in red, thin filaments in blue, and then a supporting cast of structural proteins holding everything together. If you want to skip the rest of this video, all you need to know is that the thick filaments power muscle contraction by using all these little heads to grab and pull the thin filaments down like this, thereby making the whole sarcomere contract lengthwise. But come on, how does that even work? Let's keep going. The middle of the sarcomere is, get this, the M band. This is also the dead center of these thick filaments and their mesh of support proteins. Basically, two identical thick filaments sprout out of this spot, facing away from each other, trunk to trunk, butt to butt, soul to soul, whatever you want to say. Moving outwards, we get to the A band, probably the best defined area of the sarcomere, where the thick and thin filaments overlap with each other, and honestly, this is where the magic happens. But hold on, moving a little further out is the I band, which is just the thin filaments and the elastic connector proteins holding everything together, which eventually gives way to another super dense region. Behold, the Z disc, the border between two sarcomeres. Here, a bunch of molecular scaffolding glues the thin filaments of two neighboring sarcomeres together, while providing an anchor for the elastic connectors of both adjacent sarcomeres as well. Now, this whole thing has been a glorified two-dimensional side view. If we look at this thing from the bottom up, we can see that a bunch of thick and thin filaments are organized around each other in kind of like repeatable layers. Because of how elastic and pliable our muscles have to be, the details here are kind of fuzzy, but at the basic level, these muscle filaments are arranged in this repeating hexagonal pattern. Every thick filament is more or less 45 nanometers away from the thick filament next door, while the thin filaments fill in the gaps. Alright, cool, that's the lay of the land, nice. We've got the shape of things down pretty good, but how does all this work? That's kind of like why we're here, right? And you know what? Surprise, we're actually going to have to go down one more level and break these filaments into their component parts, which is going to tell us everything we need to know about how your muscles contract. But first, a quick word from the advertiser for this video, which is surprise me. Today's episode is brought to you by my supporters over at patreon.com/clockworkshow. We've been developing a new benefit model with physical perks for the last 6 months over there, and the response has just been incredible. If you like videos like this, they really only are possible because of the support I get from my patrons. In return, I'm able to send genuinely beautiful stickers, temporary tattoos, and paper crafts to my audience around the world. It is such a joy to get to make beautiful things for such beautiful people, and these perks are only going to get better in 2026. We're really close to being able to take some really big swings here at Clockwork HQ, and it would mean the world to me if you checked us out over there. Folks who join us at patreon.com/clockworkshow by clicking the link in the description will get last month's perks as a free bonus, so you can have all these amazing sarcomere stickers and paper crafts on top of watching this video. If you thought about joining us before, now really is the time as we enter into a make or break period here at Clockwork HQ. But anyway, let's get back to building this sarcomere. Here we are, where we can finally unpack the mechanics of how sarcomeres work. Again, at a high level, these structures contract when the myosin heads on the thick filament pull the thin filament toward the M line. This system has been fine-tuned over the last billion years or so. I mean, both actin and myosin are ancient proteins that go back to the dawn of eukaryotic life and our days as single-celled blobs floating around the ocean. And coming up, we're going to see how these individual proteins create an incredible system for so much motion. So let's break it down. First up is the thin filaments. They basically hold the whole sarcomere together and are like the medium that's being contracted throughout your muscles. The star of the show here is in blue. It's the versatile actin protein that's pretty much ubiquitous across all life. Actin is a wonder. Your cells are constantly pumping out these little subunits, and when activated, they can spontaneously polymerize into these cool little filaments. And even cooler, they can also spontaneously depolymerize on the back end as well. As the title implies, actin is from your cytoskeleton, so it's going to provide some structure and some rigidity throughout your cells, but your body also uses actin's ability to polymerize and depolymerize on demand as a way for folks like your white blood cells to move around. And I mean, look at them go, right? All those little, tentacles, right? They're probably membrane protrusions being driven by actin polymerization. This stuff is incredibly fast, but the role actin has in muscles is a little different. We want our actin here to stick around and polymerize in the right way at the exact right length and not fall apart at random. So your muscle cells are going to build some support structures around actin to make it a filament and not something super dynamic that breaks apart too much. First up, your muscle cells build this little yardstick called nebulin. Nebulin doesn't look like much, but it's one of the single largest proteins your body builds on a regular basis. And if that makes you ask what the biggest one is, sit tight, that's coming up in seconds, right? Nebulin's job is to act as a scaffold for actin to polymerize around. Instead of letting actin get all dynamic and depolymerize on a whim, nebulin stabilizes actin in its filament form, keeping your muscles from breaking apart for no reason. This gives nebulin the power to determine sarcomere length, but more on that towards the end of the video, actually. And the thin filament is further stabilized by this coil, tropomyosin. And sure enough, tropomyosin also has a really important job outside of stability, working in tandem with this last protein called troponin. But let's build the rest of this sarcomere before we get ahead of ourselves. Before we head down to the thick filament and the main event of this video, really, I may as well mention what is objectively the coolest protein in your muscles, since it's also up here anyway. These cables are copies of the same protein, titin, which is incredible in its sheer mass. What you're seeing right now is actually 3 copies of titin that surround a single thick filament. A single titin chain is one singular protein coded for by the single longest gene in your DNA. Titin in our muscle cells is 34,300-ish amino acids long, just incredibly massive. By comparison, a more average protein like hemoglobin is only about 550 amino acids when you add up all the subunits together. So we are on just an incredibly massive scale. And you know what, quick tangent, I was gonna make a whole thing out of the fact that Titin is the largest peptide known to science, and how cool that is that it's, in vertebrate muscles, but then, 2 years back, this team out of the Scripps Institute in San Diego ruined the fun by uncovering an even more massive protein, PKZilla, that's 30% more massive than Titin while also somehow being cooler, and uses all that unmodelable heft to help this one algae build this really intense-looking toxin or whatever. I mean, honestly, that just kind of feels like its own video, though. But anyway, don't let Titan's outsized reputation intimidate you. This structure is manageable once you peek under the hood, because instead of being like 34,000 distinct little puzzle pieces making up this cable, it's basically just the same 3 protein domains repeated a bunch of times. And to be even more real with you, it's just these immunoglobulin and fibronectin domains that get copy-pasted like a few hundred times while Titan is synthesized. The fibronectin domains provide rigidity and structure, while up in the I-band, these immunoglobulin units fold and unfold, allowing Titan to act kind of like a molecular spring, absorbing some of the tension and force from the stress of contraction. Titan spans the whole length of the sarcomere, with a single Titan cable anchored to the M-line and Z-disc. The tension and elasticity that Titan brings to the table helps hold the sarcomere together, and that spring action allows Titan to help pull the sarcomere back to the relaxed state after muscle contraction, which gives our motion a ton more power and flexibility compared to folks like insects and other critters who lack Titan in their muscles. But getting back down to the biochem real quick, what's cool is that these links actually provide elasticity in a few different ways. First up, the actual individual links in these chains will bend together a little like this when your muscles are relaxed, and then these kinks will straighten out as your muscle contracts more and more. And this is where it gets wild, because according to certain models, some of these immunoglobulin domains can unfold completely into a straight line when the cable is under enough tension, which makes Titan even springier. Our bodies can use these different levels of elasticity to accomplish so much extra work, but I'll need to save those for a part 2 where we improve this sarcomere model with better simulations of how Titan works. Make sure you comment if that deep level of biophysics is something you actually want to see. I mean, that's the only way I'll be able to put myself through the pain of that level of modeling. Anyway, though, for now, just remember that Titan is a long elastic cable that has a couple of distinct regions where these immunoglobulin and fibronectin domains will repeat in a certain pattern. That's going to be important to have in mind when we round things out by making it all the way down to the thick filament. Which, speaking of which, let's just get down there now. The myosin filament. Finally, we're getting to the meat of this thing. The core of your muscles are built around these thick filaments, which mainly are a rotating stack of these long myosin molecules. Our muscular myosins come in pairs with long, winding tails that wrap around each other, which are both capped off by a motor domain attached to a flexible neck. These myosin domains kind of helically stack on top of each other, which is how we build our thick filament. The average myosin filament is going to have around 294 of these myosin molecules stacked on top of each other, with 2 kind of identical copies branching out trunk to trunk from the M-line. There's one last super cool detail about how this all comes together, but I'm going to save that for the end. For now, let's get to the main event. Let's see how these myosin motors pull on the actins inside that thin filament. And to do this, let's get out of this big simulation and zoom in on the interaction between one myosin and a nearby thin filament. What we're about to see is the power stroke cycle for these myosin motors. Stick with me here, we're just going to level up through it. Ready? So how exactly does myosin bind and move these long, thin filaments? As always, it comes down to one of the most important maxims in all biology: structure equals function. And myosin is able to pull on actin thanks to a repeated sequence of very subtle shape changes. Shape changes that are powered by everyone's favorite energy currency, adenosine triphosphate, or ATP. And you know what? We're just going to start at the very end of the last power stroke. Myosin has just finished pulling this actin molecule up a little bit, but it's still bonded to this actin unit. We really don't want to stay stuck like this for long because we don't want to jam up the rest of the sarcomere as the muscle continues to contract. Luckily, the mitochondria that are crammed up against these sarcomeres are just flooding this area with ATP, which means a fresh energetic molecule of the stuff is going to quickly find and bind with the little pocket on the back of our spent myosin right here. And this is one of the magic pieces of biochemistry. Almost every single thing that happens to any protein is going to change its shape a little bit. That's how dynamic things are down here. And therefore, even the little nudge from this ATP is going to change the shape of our myosin head just subtly enough to help it dislodge from this actin filament, freeing it up to start the next power stroke cycle. And this is the cool part that makes the power stroke so automatic and so fast. Letting go of this actin is going to release a little tension inside this motor. This conformation is perfectly primed to hydrolyze this ATP. This terminal phosphate group at the end will pop off, which, to oversimplify, releases a burst of energy that the surrounding myosin motor domain can use to change shape. And myosin can only move one way when charged by an ATP burst like this. That energy will throw myosin's lever arm into the primed position. That burst of energy is going to cock the myosin domain back down to the prime state, readying it to grab onto the next actin molecule down the chain. And this brings us to the real magic of myosin and the secret sauce that makes your muscles work. You hear about ATP in biology all the time. ATP is constantly moving energy around your cells by getting hydrolyzed, popping this phosphate group off and releasing that burst of energy. For a lot of proteins, ATP slots into a binding pocket like the one inside myosin right here, transfers its energy, and is then immediately released. But in myosin, when ATP cocks the motor back, it actually gets stuck in the binding pocket here. Sure, this myosin head is now under a lot of tension, but that tension can't get released because this adenosine diphosphate, ADP, and the phosphate it just released are jamming the works up in here. Maybe this sounds clumsy, but it's a brilliant way to make sure that these myosins don't burn through cellular energy for no reason. Instead, think of this like, I don't know, a mousetrap. We're effectively storing the energy from ATP until the exact moment we need it, which is when this myosin comes in actual physical contact with an actin. There's a great binding spot for myosin in this groove on actin right here, and attaching to that actin will change the motor domain's head just enough to pop out this little phosphate, which again allows all that tension to kick the myosin lever arm here down, which for physics reasons is going to actually shove the motor and actin up, which is the actual power stroke of this cycle. As soon as the power stroke here finishes, a new ATP can slot in and free myosin from its bonds to actin and start the cycle all over again. And this system by itself is incredibly powerful and beyond versatile. This automatic interaction between actin and myosin powers cellular shape changes, cell division, some motor proteins, and a whole host of other jobs all throughout the whole eukaryotic domain. Now yes, this is complicated, but the complexity here allows for one last incredible layer of control. Because if binding to actin automatically activates these myosin motors, and, actin is kept within binding distance of myosin basically at all times in these near crystalline sarcomeres, then, how do our cells keep myosin from just power stroking all the time? I kind of wish I had reworded that, but I'm in too deep now, so let's just keep going. Because this is the best part of the video. This rocks. We get to put the whole thing together now. Let's focus back on the thin filament and check in on the last piece of this puzzle. Remember, our thin filaments aren't just raw actin here. They're stabilized by these winding coils of tropomyosin. Actually, coiled coils of tropomyosin, but whatever. And those coils are occasionally studded with these troponin complexes we glossed over earlier. Now, troponin controls the real magic of the sarcomere, and here's how. Turns out, tropomyosin isn't just stabilizing our thin filaments, it's also actively blocking the part of the actin molecule that myosin can bind to. During inactivity, these myosin motors are just endlessly teased, forever caught in that post-ATP tension with nowhere to bind. All tension, no release, until you actually decide it's time to flex something. When it's actually time for a muscle to contract, your nervous system is going to send an impulse directly to that muscle. That nerve contact is directly connected to all those little tubes that surround the myofibrils we discussed earlier. And once that signal arrives, the sarcoplasmic reticulum here turns on the pumps and floods the myofibril with calcium ions. Once inside the sarcomeric space, that calcium will bind to troponin, which will activate and change its shape pretty sharply. That shape change is going to pull tropomyosin away from actin's binding cleft, allowing myosin to actually contact the filament and roll through the power stroke cycle. Meanwhile, at the exact same time, there are pumps between the sarcomere and the sarcoplasmic reticulum that are constantly pumping calcium out of the myofibril, which keep the, contract signal short and to the point. But yeah, that's the main gist of it. Vertebrates like us have re-engineered cytoskeletal proteins to massively increase the amount of leverage we have over the outside world. For me, the most important thing that allows this to work is how perfectly organized these sarcomeres are. Don't forget how they line up so well to ensure that all these little proteins end up all working in the same direction. How do we even pull that off when living structures can look so messy in other ways? And this is actually another one of the superpowers of Titan. Remember how Titan is just 3 different subunits repeated over and over in different patterns. Down here in the thick filament, Titan just repeats these immunoglobulin and fibronectin domains in an 11-unit pattern over and over again. And this 11-unit pattern actually repeats 11 times. Nice. This is the C-type super repeat, and evidence suggests that this acts like a molecular ruler, giving the tails of these big ol' myosin molecules a place to start binding. Once that first myosin molecule slots in on this first super repeat from Titan, the rest of the myosins can slot into place, stacking up helically like this, all 294 of them stacked helically on top of each other, and the rest of the filaments fall into line all around it. With Titan and Nebulin, we added a few extra yardsticks and controls, and that's going to help the fuzzy process of cellular life achieve a kind of incredible rigidity and specificity. For me, it's incredible how all this comes together. I love so much how our cellular structures have relatively simple rules that build to fabulous complexity. And like I said at the very beginning, this system is also incredibly modular. By organizing the sarcomere around yardsticks like Nebulin and Titan, our cells can make incredibly different muscle types out of pretty much the same molecular ingredients. If you've seen my big ol' video on how DNA gets transcribed into RNA, you know that one of our secret superpowers is RNA splicing. This big complex called the spliceosome modifies RNA as it gets transcribed from DNA, allowing us to produce tons of different proteins from a single DNA sequence. Alternate splicing of Nebulin and Titan changes the length of these yardsticks and therefore the length of the entire sarcomere. Different lengths and a few other modifications allow for a breathtaking variety in muscle types and jobs. A little added complexity yields an astonishing amount of extra control. And gosh, we are able to do just so much with our own internal variety. That's the main thing I always want you to take away from videos like this. Together, we slog through a whole mess of dense information, and in the end, we're able to put things all together and see the majesty in our biochemical foundations. Frankly, muscle cells are great in that these sarcomeres are organized enough that it's easier for me to show you just how jam-packed our cells are with proteins and structure. It's crowded in these cells, and we're able to do just so much with all these little puzzle pieces. And there's so much more to explore in these sarcomeres, but here's a good place to end this first video. Make sure you hit that comment section if you want to dive even deeper into this particular aspect of chemistry. For now, though, I really appreciate your time. Thank you so much for watching. And hey, thanks so much for getting all the way to the end of this video. If you liked this video, make sure you like and subscribe so you don't miss future editions of this series on muscle structure. At the same time, it would mean the world to me if you checked out what we've got cooking over at patreon.com/clockworkshow. This year has a lot of great perks in the works, and I think you're gonna really like it over there. And one bit of housekeeping, I just want y'all to keep in mind that so much got left on the cutting room floor here too. I didn't get to talk about creatine or low duty ratios or anything cool like that. So make sure you let me know if that's something you'll really want to see. Otherwise, the main thing I want you to know is how much it helps me when you make it all the way through a video like this. Just the act of you hearing these words right now effectively is carrying this channel. Thank you so much for being here. But that's a solid place to end things. Let me know what your favorite color is in the comments if you made it this far. For now, though, I just really appreciate your time. And as always, I like to leave you with peace, love, and as many C-type super repeats as possible. Everyone be well. Thank you so much.